Hello, my name is Alexander Katko, and I'm an immunology PhD student, a part of the Morality Lab. My research focuses on the maintenance of immunological memory over long periods of time. So, immunological memory is induced by repeated exposure to the same antigen and allows the organism to respond much more rapidly and efficiently to that antigen. For example, memory cells can start secreting cytokine within minutes of re-exposure as compared to hours or days for naive cells. The hypothesis of this work is that this memory is maintained over time through changes in chromatin accessibility and transcription factor binding. Previous work has demonstrated that memory is correlated with the priming of genomic regulatory elements proximal to rapid recall genes. Rapid recall genes are genes that are quickly turned on in memory cells, but aren't rapidly upregulated or downregulated in naive cells. So the goal is to create transcriptional regulatory networks using human CD4 positive T cell multiome data. Multiome data includes paired RNA and attack-seq on each cell simultaneously, and the resulting TRN can be used to model and predict transcription factors that maintain these poised regulatory elements. The experimental design includes 4 human donors. Peripheral blood was collected from each of these donors, and 3 major CD4 positive T cell populations were sorted for and enriched: naive cells, T effector memory cells, and T central memory cells. TCR stimulation was then performed using anti-CD3 CD28 beads to obtain 4 conditions: resting, 2.5 hours, 5 hours, and 15 hours. A unique part of this experimental design is the inclusion of a 4th spike-in donor. CD4 positive T cells were enriched using the same method as the experimental donors, but no TCR stimulation was performed. Cells from the control donor were spiked into each group and will be used to help differentiate between batch effect and true biological signal. One computational challenge was tracing back which cell came from the control group and which came from the 4 experimental donors. Whole genome sequencing was available for the control and inferred for the experimental donor using the multiome data. Supercell was then used to cluster cells based on genomic variants. Cells were then labeled either as control or donor. And here is a brief remark on transcriptional regulatory network inference. So gene expression can be modeled as a multivariate linear model of transcription factor activity, or TFA. TFA quantifies how each TF regulates its target genes. The inferolator is the method we use for TRN inference. The inferolator calculates TFA using both gene expression data and prior information on TF gene interactions in the form of single-cell attack data. So as you can see, this is why multiome data is so good for TRN inference. The inferolator results in a ranked list of TF gene interactions that are supported by the attack prior, but retains the ability to learn new interactions. At each time point, the multiome data was integrated by donor to ensure human donor was not a major source of biological variation. The data was reduced in dimension using UMAP, and cell types were labeled based on gene expression. For each of the time points, different populations of resting naive, resting memory, activated naive, and activated memory were delineated. Then pseudobulking was done, where each population is treated as one cell and the gene expression added up and normalized. A clustered heat map of gene expression by cell population is displayed in the lower middle of the poster. One observation here is that naive resting and naive memory cells have very similar gene expression, but very early on after TCR stimulation there is a divergence in which different genes start to be up and down regulated. Finally, I'd like to share some early results from the transcriptional regulatory networks that were generated. These results are displayed in the upper right-hand panel. The first 2 heat maps are so-called core TFs, or transcription factors that are important to a particular cell type. So each cell type has a group of differentially expressed genes, either up or down, and a particular TF is a core TF for that cell type if its edges are enriched in this set of differentially expressed genes. It's clear from these heat maps that resting naive and resting memory cells have very similar core TFs, but after activation, there is a divergence in which TFs are important. Many of these transcription factors make sense from what we know about immunology. For example, one major core TF for the resting cells is KLF2, which is known to be important for CD4 T cell survival. The heat map on the left corresponds to core activator TFs, and in the middle are core repressor TFs. It is of course possible for a core TF to be both an activator and repressor depending on its target genes. The third heat map in the row is the TFA matrix, which is displaying each transcription factor's activity for each cell type. Finally, in the bottom right, I'm showing you a TF-TF module analysis, which are transcription factors that work together. And by working together, what I mean is that there is a significant overlap between its target genes. And once again, we see many expected clusters of TFs, such as a group composed of the IRF transcription factors. I hope this has helped make clear what kind of results we are capable of producing using these transcriptional regulatory networks, and I have a lot of more ideas on other things I'd like to use them for. So thank you so much for listening, and if you have any suggestions or comments, please feel free to reach out and contact me.